Good afternoon, everyone. I'd like to start this session entitled, session number 33, entitled Unlocking Barriers and Access Inequities to Museum Collections in Archaeology and Paleontology. And I am one of the session moderators, and my name is Takumi Tsutaya, and the other co-moderator is Yuri Kimura here. So yeah, I'd like to start from my own presentation. So yeah, this is my presentation. So yes. First, I'd like to introduce the theme of this session. This session aims to explore the balance between preservation and management of specimens and destructive analysis in academic fields that study the past, such as archaeology and paleontology. The objective is not to solve a specific problem, but rather to allow researchers involved in this specimen management and analysis to share their cases and thoughts, fostering a brainstorming-like atmosphere to deepen our understanding of this topic. There are 4 presenters in this session study the past, which probably differs from the core members of Spinach and Tadwick. By integrating these different perspectives, we hope to generate new ideas and synthesis of an innovative framework. After the 4 presentation, I hope we can have a time for discussion, and yeah, comments and contribution from the audience are highly appreciated. So in my presentation, as a researcher of ancient biomolecules, I will review the trends in destructive analysis in archaeology and paleontology, and examine the issues in each processes. This is an introductory presentation for those who may not be familiar with this field. Typically, destructive analysis in archaeology and paleontology follows this flow. First, specimens are excavated and collected by institutions such as universities or museums, where curators identify, register, and store the specimens. Researchers who perform analysis need to access these stored specimens. After obtaining official permission to conduct research, they sample a portion of specimens. The subsamples obtained are then analyzed in specialized laboratories using data. These results are typically published in scientific papers. After the project is completed, any remaining samples are returned to the curating institution. Finally, the results are contextualized and disseminated to the broader public. The main issues of these processes are shown in red on the right side of this slide. Here, I will summarize these issues in detail. Archaeological and paleontological excavations are labor-intensive at first and require highly specialized skills. Furthermore, archaeological and paleontological specimens are diverse, including invertebrates, vertebrates, plant remains, or human artifacts such as potteries or stone tools. As a result, different researchers or research groups often conduct excavation, identification, and analysis, leading to the involvement of numerous stakeholders with varying expectations and demands. The presence of such diverse stakeholders is one of the factors creating various tensions in destructive analysis. Also, sectors outside academia are also significant stakeholders in this field. Citizen scientists are essential to archaeology and paleontology, but they are also bringing accompanying issues. Regarding specimen collection, particularly in paleontology, commercial trades are common. However, this also means that researchers in wealthy countries have easier access to fossils. Additionally, when fossils are collected as a hobby or financial investments, they often become unavailable for research. In the case of archaeological artifacts, looting can result in the loss of context, such as stratigraphy and associated materials at the time of excavation. Looted artifacts become just an art object and not used for research. The recent financial situation surrounding museums is harsh, as you know, with some museums auctioning off their collections to private collectors. To secure spaces for new collections, some museums take shocking measures, such as reducing existing specimens or selectively accepting only a portion of new specimens. Most researchers do not want to talk about the disposal of specimens. However, researchers who have published papers on the reduction of archaeological specimens are concerned that when the time comes to reduce specimens, we do not have a theoretically developed effective method. Also in Japan, my research collaborator, the Osaka City Cultural Properties Association, will be disbanded due to a top-down policy shift. A vast number of archaeological artifacts will be locked away in storage without proper management, with no electricity or water supply. Regarding specimen access, issues of colonialism and repatriation are the major topics today. There is a lot of previous research and debates on these topics. Repatriation is now expanding from archaeological human remains to fossils. One of the most notable cases is a valuable feathered dinosaur fossil originating from Brazil. In this case, a related paper was withdrawn and the specimen was returned from Germany to Brazil. As a distinct feature of archaeology and paleontology, every specimen is unique and cannot be renewable. The advancement of analytical techniques is so rapid, and there is always a possibility of the development of more sophisticated and effective methods with smaller destruction in the future. Thus, destructive analysis in archaeology and paleontology constantly faces the dilemma of whether to conduct destructive analysis now to obtain immediate results or to preserve the specimens for long-term use with advanced techniques in the future. One such issue is the petrous bone. The petrous bone is the bone around the inner ear and is known to preserve ancient DNA better than any other skeletal parts. As a result, petrous bones have been increasingly sampled worldwide in recent ancient DNA analysis. However, sampling destroys the morphologically important inner ear structure. That raises concerns about the declined availability of petrous bones for various future research. Furthermore, ancient biomolecular analysis is highly capital intensive. Consequently, only a few groups worldwide could conduct full-scale ancient genomic analysis until a few years ago, leading to oligopoly. Research attainments, career opportunities, and fundings are concentrated in these groups, creating distorted incentives even for specimen managers. There is also a question of who owns the specimen. In practical terms, attitudes to the byproducts and leftover samples from destructive analysis vary from person to person. Moreover, when an analysis is conducted but no ancient biomolecules can be retrieved, such information is not often reflected in the specimen database. To prevent unnecessary destruction in the future, this is certainly a negative aspect. Furthermore, a debate occurred last year in paleoanthropology. A researcher sent a few million-year-old hominin bones from South Africa into space. Many researchers criticized this, as sending bones into the space had no scientific significance and was just self-promotion that reminded us of colonialism. This topic raised questions about who owns the specimens and for what purpose they can be used. Finally, archaeology and paleontology attract one of the most intense public interests. Everywhere in the world, children are fascinated by dinosaurs, for example. Such widespread interests can foster public understanding, but it also creates issues such as the gap between the commercial and academic sectors. For example, the field of ancient DNA has developed along with the growing interest from general public, fueled by the popularity of commercial media like Jurassic Park. To summarize, specimens are unique and non-renewable in archaeology and paleontology, and because excavators, curators, and analytical researchers are often different persons, there is a constant tension between preservation and destruction. This tension has been further exaggerated by recent publication pressures. Additionally, because of the high level of public interest, the tension between preservation and destruction is also significantly influenced by social factors. Okay, this is all. Thank you so much. And I would like to accept one or two questions from the audience. Maybe no? Okay, so we will have a general discussion time after the 4 talks. So we would like to proceed with next presentation from Ito-san.
Good afternoon, everyone. I'm Yasuhiro Ito. I belong to the Kyushu University Museum, Fukuoka, southwestern Japan. I'm a collection curator of paleontology. Today, what I want to tell you is that we will link the specimen data. The basis of specimen data is, of course, the specimen. When the specimen data is linked, the specimen is exposed to the public, and the accessibility increases. And consequently, it leads to conservation of the specimens. So we will document digital data from the specimens and link the specimen data. Furthermore, we believe that making specimen data public is one way to achieve the theme of this session, which is unlocking barriers and access inequities to museum collections in archaeology and paleontology. We will introduce 3 our activities to link the paleontological specimen data. First, cross-searching WebDB, a web database. In Japan, each museum has its own web database. That is almost native language Japanese. sorry, we don't own the copyright. This is an essential service for students, local people. However, these are in different data format and different database systems. So first of all, it is important to integrate these databases. The integrated database is the Japan Paleobiology database. Database that is the core of our project. It's called JPaleoDB. This is a portal site to collectively search paleontological specimen data in web databases published from museums throughout Japan. This is a basic function of this site. When you search as a result, a table is displayed. Then each result row links directly to each specimen page on each museum or institution, WebDB. Well, very simple system. Cross-searching WebDB, the search result of JPaleoDB links to the museum WebDBs all over Japan. In other words, JPaleoDB enables one-stop search for Japan's paleontological collections. Second activity, WebDB publishing. So, many museums manage specimens using digital data, such as Microsoft Excel files, without publishing on the web. At these museums, they want to release specimen data, but do not have the means to publish the database. In this case, we help to publish WebDB. And the specimen data will be integrated to JPaleoDB. Now, 50 museums contribute in Japan. Most of the digital data of paleontological specimens are integrated in JPaleoDB. JPaleoDB enables one-stop search for these museums. The data is about 430,000, holotypes 7,000, vouchers more than 7,000, voucher specimens described in papers over 100,000. Paleontological publications of in Japan over 16,000. Now, cross-searching WebDB and WebDB publishing. These two activities are basically about already curated specimens. So these activities can be done with personal computers. On the other hand, about the uncurated specimens, we cannot do anything on the computer. In Japan, many fossils have been dug up by civil engineering work, road, bridge, dam, and tunnel construction. Many of the collected fossils are stored in the regional museums. These fossils tell the geological history in the region. Some famous fossils have been studied, for example, vertebrates, such as dinosaurs, whales, elephants. But many other specimens, for example, shell fossils, are just stored without curation. Third activity, curatorial support. In order to link to these hidden specimens, we must curate and digitize them. We will introduce one case study about curatorial support that helps curating paleontological specimens in the museum. This is the Kitami Region Museum. This is a map of Japan. Okinawa is here, southern tip. Kitami is in eastern Hokkaido, northern tip. Here, Fukuoka, Tokyo. This museum houses many fossil specimens collected from the region. But there is no paleontologist. Museum curators are archaeologists. And specimen curation. We used to shell fossils collected at dam construction. These were collected in 1994. They have been stored for 25 years without curation. We curated these fossils at the workshop in paleontology for curators in eastern Hokkaido. They all specialize in fields different from paleontology. So, beginning with paleontology lectures, specimen preparation. Before and after. This is the result of the workshop. Before, specimens were remained collected. After, specimens were sorted by fossil taxa, and specimen cards were attached. Result. Basic identification data were listed, and we can ask as specialists for identification. Left side, left row, species name are more detailed and modified. By the way, this picture is identifying plant fossils, another curatorial support. And the WebDB was also built. Of course, the WebDB is integrated in JPaleoDB. Through this curatorial support, we are increasing access to uncurated hidden specimens. Well, through 3 activities, we are promoting a project to link the paleontological specimens. To end, we have introduced our activities. Anyway, it is important to curate, digitize, and link the specimen data. Currently, our specimen data is mainly native language Japanese. But mass data collected in our project can be efficiently converted to Darwin Core. Once the data is converted to Darwin Core, the data is shared with many global databases. So Our project also links regional museums and the world. Thank you so much.
So we have 30 seconds. Just a quick question. No? Okay. Thank you so much. So next, please, Kimura-san.
All right, so we are a group of paleontologists and anthropologists, and my talk, and our talk, also three of us, are seeking balance between long-term care and district analysis in paleontological and anthropological collections at our museum, National Museum of Nature and Science in Japan. And two of our presenters are from this museum, and as a curator, we manage three kinds of jobs and in a collection we are in charge of. And the fact that we are both collection managers / researchers is kind of important in this talk. In a typical scene of paleontological and anthropological investigations, bones are unearthed during excavation campaign, and these materials are carefully prepared and tagged with site information and age and other attributes. They are stored in a museum with unique IDs, and scientists come to the museum to do the research on these specimens. And when geochemical analysis and DNA extraction became more popular in these fields, and things started changing in our museums. And these analyses are almost exclusively destructive. In other words, the aliquot is removed from specimens to obtain valuable results. So let's imagine how the specimen 007 is processed in a study and how it obtains new attributes. It starts with, say, MNS 007. The aliquot specimen is also the same 007, the same number, but this piece would get a new ID for lab management. And if not all, some portion of 007 piece would be ground up and go through the further extraction processes. The resultant data may be stored in a proper online database, and it has its own ID as well. In the whole process, the researcher would know precisely where the original specimens are located and can track the entire process backward, but for museums, not so much. So here I want to introduce an actual case, which I'm not quite proud of. A few years ago, I was contacted by a researcher group of ancient DNA from Canada, and they're trying to obtain samples from the fossil elephants. And the Paleoxydonomani, the endemic species of Japan, was one of their list. And I used a very typical long format to loan those samples, not really giving the sample to them. The portion of PV23905 probably went through the same process I just explained, but still I haven't received any information since the specimens has arrived to their lab. And there seems to be a glass wall on the museum side. And if all specimens are used in analysis, then probably the story ends there. But however, I know that in many cases, the leftover samples are kept in the vials, and then probably half of the aliquot is analyzed, but the other half is left intact. So who owns the sample, the leftover sample? It really depends on the agreement between the researcher and the museum. However, there must be operational obstacles in terms of long-term care for sensitive materials, assuming that conventional storage is available in the museum. The processed sample might not be suitable to be returned back to the museum. The trouble I came across is also related to the ambiguous procedure in destructive sampling requests for paleo collection, probably anthropology collections as well. If you Google keywords of DNA request form a museum, then you can easily find the request form of major museums for extant materials. Nevertheless, there's no standard request form for paleo collections. Do not exist as far as I know. And considering the rarity of fossils and artifacts, these requests must be examined more carefully and before those destructive samplings is approved. So here, I want to introduce 4 cases in which destructive sampling was approved in our museum. Specimens of a stegosaurus was studied to examine how the plates and the spikes functioned in these dinosaurs, and a thin section of the plate and the spike was made to observe internal bone structures. The sampling of the specimens allowed with conditions that the studied sections were returned back to the museum, and when the research was completed. So now it is back to the museum. And then these samples are adequate. It can be considered as an adequate of the samples, and there is no specific ID is given to these thin sections. And you can also notice that the destructive parts of originals here, and it's not reconstructed. And this is the second case, and this is a neck bone of a Paleontological anomaly, a very endemic elephant that used to live in Japan. And this was sampled for radiocarbon dating, and the bone powder were collected by drilling here. And then the drilled hole was actually filled with some sort of plaster materials by the researcher group. And then I also want to emphasize that the replica of these fossils was not made because the drilling portion is much smaller than the entire fossils. When the original fossil is very tiny, we ask for high-resolution cast with epoxy resins, and these small tests were performed for stable isotope analysis, and this was actually done by my own research. And after the completion of research, these samples were returned to the Peabody Museum at Harvard University, which has a long-term agreement with Geological Survey of Pakistan. And I made 3 sets of casts for each institution, my museum and Harvard, and also the Pakistan Geological Survey. And some fossils are so rare because their locality is already gone or very hard to access. And this bison tooth, and also from Japan, came from such locality. And this is actually an ongoing project by Takumi and his colleagues, including me. And considering the rarity of this locality, we made a fossil replica before he's going to correct the ancient DNA sampling. And to save time, we chose to preserve tooth morphology by CT scanning, and a subsequent 3D printed replica, replica was made. And the tooth is currently loaned to Takumi and his colleague, but adequate of these samples will be returned to our museum after his research is done, and the other extracted material, if present, will stay in his lab for the sample properties. So it is very important for us to keep a record of what analyses have been done on a given specimens and where the sampled material of the specimen is stored. And we recently added these attributes in our database, and the database managed with our own language, Japanese, but the collection location was added to the database, and I hope to add another attribute to tell what kind of analysis is done on those specimens. So it is very important to keep those on the database. That's what I learned from the issue that I encountered with ancient DNA groups. And then sometimes, and I want to emphasize that sometimes the rarity of the fossil increase as the paleontological research proceeds. So let's go back to the paleontology and the Paleoxton specimens at 23905-965. The species status was not known when I got requested from the ancient DNA group. But I know the location of this fossil is quite unique. It was found a boundary between estimated distribution of a Taiwanese Paleoxodon called the Huaensis, and then other distribution by Paleoxodon anomaly. So it's in the boundary, and my student Mao was very interested to know which species this fossils belongs to. So Mao conducted the multivariate morphological analysis for her senior thesis. And she still have to get lots of data before she make any conclusion. But based on her limited data, she suggested that the specimens should be assigned to Paleoxodon anomalies, so Japanese species. And if this is true, this individual, it must be the largest Paleoxodon ever known. And this guy probably roamed in a Pleistocene gateway to Japan and before Dolphism started. So this is suddenly very important specimens. So based on what she found so far, it would be ideal to do an independent age estimation, or get the specimen's age. And that requires another destructive sampling. But she already proved that the specimen is one of a kind, and I prefer not to destroy the specimens again for new samplings. So if the aliquot of the specimen is kept in the DNA group, it would be ideal to use their samples. So a lesson I learned from this issue is that trackability is really important for the rare specimens like paleontology and anthropology collections. So I really want to, and we are very glad to be in this group, in this joint conference for the very first time as a paleontologist and archaeologist. And you guys have more knowledge, advanced knowledge about keeping all information from the specimens. So please share us your experience in this group, and hopefully we get some idea and include it in our paleo collections. So thank you very much.
Thank you. We have 2 minutes remaining, so do you have questions?
Thank you, Yuri, very much. This is very much needed for our discipline, a great conversation starter.
Thank you.
Have you also considered documentation of applied materials in a preparation lab to given specimens, and how that factors into dissemination to researchers who are interpreting results, such as radiocarbon dating?
I mean, documentation on
applied materials like adhesives and consolidants and other things that may have been used in molding that would perhaps introduce younger carbons and influence the interpretation of results that are destructive analysis based.
Yeah, so we are very much behind all of them, and then you really have to prepare. It's actually a good idea to make some documentation about the protocols. You were talking about the protocols, right? Yeah, it's very important. Thank you so much. Yeah.
Other questions? Okay. Thank you so
All right,
much.
thank you so much. Yeah.
So the fourth presenter is Masaki Fujita. Yes, please.
Yeah. Hello, everyone. My name is Masaki Fujita, working in the National Museum of Nature and Science. My background is anthropology and doing some archaeological excavation, mainly in Okinawa and several other places in Japan. So today I will talk about importance of the multidisciplinary analysis at archaeological sites before storing the remains in the museums. The destructive analysis is a relatively new technique, and it is a very powerful technique for unraveling the prehistoric human life. But it is a very difficult problem for museum staffs, because we have the mission of preserving specimens and passing them on to future generations. So in our department, the Department of Anthropology in the National Museum of Nature and Science, we have no destructive analysis is prohibited in principle, and no approval criteria we have for destructive use of the specimen. Because if we set the criteria for the destructive use of the specimen, we would have to accept all researchers that meet the criteria. We know that the destructive new technique is very important for developing the academic field, but it is difficult for us. and we also know that each researchers need only small amount of the sample, but how many researchers we were able to accept, or who is the best person, first requesters. Yeah, that is a very big problem. We do not have any good fundamental solution at present. So I recommend, my recommendation today is that the analytic destructive analysis should be applied for the specimens before become a museum collection. As Tsutaya-san told, we have in the case of the archaeological and anthropological samples, they are firstly excavated in the field and then brought to the laboratory and cleaned, sorted, and reconstructed, and the basic researches have done. And after that, we make a database and registered in storage. So, and we usually spend several years at least in the case of academic research. And we have many variety of materials we have especially at the excavation field. For example, human bones, animal bones, and plant remains, and not only those organic materials, but also stones, minerals, soils, and also various artifacts. And we excavators usually consider that our activity, excavation itself, is activity destroying the archaeological site. So we want to know as much information as possible through the excavation. So any kind of analysis were welcome at the excavation site. So I myself is conducting several archaeological research and asked my collaborators to do the destructive analysis. So I would like to introduce several case studies. And the first case is the Tsuzupiskiabu Cave, early Holocene site in Miyako Island in Okinawa. In this cave, we obtained the many animal remains and from the early Holocene layer. Most of them are most abundant species taxa is wild boar teeth. And in addition, we have some stone tools and only 3 isolated human teeth. And my friend anthropologists have strong interest to analyze these human teeth, but it is difficult for me to accept their offer. But for the wild boar teeth, I have many, so it is easier to accept the offer. Actually, we conducted a fluorine content analysis on the wild boar samples and also extinct deer fossils in addition to the carbon-14 dating to determine the possible chronological difference between these two animals. Even though these two animals were obtained from the same layer, the fluorine content were completely different between wild boar and fossil deer. So it indicates that they originally from the different chronological age. And yeah, this kind of information is very important to considering the sedimentation process of this site. So we are very happy to know this through the destructive analysis. And the second case is the Sakite Cave in Okinawa Island. This is the Pleistocene cave site in Okinawa. And we obtained many variety of animal bones from the Pleistocene layer, Paleolithic layers. And one of the dominant taxa was the freshwater snail shells. And we asked my collaborators to do the XRD analysis and oxygen isotope analysis on the freshwater snails. The XRD analysis is used for determining the firing, cooking by the prehistoric human. We have the white colored one and dark colored one, dark colored specimens, and the shells consist of basically aragonite, but after firing, it changed to the calcite, and the black colored showed the calcite pattern, while white one shows the aragonite pattern of the XRD result. So we were able to confirm that this dark colored one was fired, burnt specimen. So it probably was consumed by the human as a food. And oxygen isotope analysis was conducted along the growth line of the snail shells. And the oxygen isotope ratio varies in relation to the water temperature. So we can get the water temperature data of this specimen experienced. And finally, that we can determine the season of the death of this specimen. So, and it means that the season of the human hunting of this specimen. And actually, we were able to know that Paleolithic people consumed these freshwater snails in autumn. Yeah, so we can get the Paleolithic human life through the oxygen isotope analysis of the snail shells. And after this study, another colleague of us tried to reconstruct the water paleo temperature of the during the last glacial maximum, and they successfully confirmed that the average temperature of the last glacial maximum period was 7 degrees cooler than today. The paleo temperature data is not directly related to the evaluation of the archaeological site, but temperature data is quite important for considering the paleoclimate and human life at this place. And we have variety of materials from the site, and we are now have ongoing project of including destructive analysis. For example, the thin section observation of the pigment obtained from this site. And we are trying to get ancient DNA from the soil sample, and also conducted a paleomagnetic analysis to determine the hearth. And also we have several burnt limestone rocks, probably used for cooking, but we are not sure. So we just start to discuss with our collaborators what kind of analysis we can conduct on these samples. Any great variety of materials we have in the excavation field, and many potential about the destructive analysis. And finally, I would like to introduce the case of using the museum collections. My collaborators did the carbon and nitrogen isotope analysis on the extinct deer fossils stored in Okinawa Prefectural Museum and Art Museum, and also the histological study on the same extinct deer collection of this museum. The reason why we were able to access this sample is because I myself excavated these fossils when I was working in this museum some 10 years ago. So now I am not working in this museum, but the researchers in this museum I have some research priority on this sample. So in summary, the joining to the excavation research, any analytical study are welcome. And so we encourage the analytic researchers to visit the archaeological site and become a member of the excavation team to apply the destructive analysis and bring the new information to the site. And not only the academic research, but we have about 9,000 archaeological rescue research in Japan every year. So there are a great opportunity to join the archaeological research. So simply I say good luck. So thank you very much for listening.
Thank you. Do you have questions? Well, thank you so much. So we have approximately 10 minutes remaining. So we chose some topics should be discussed and would like to be discussed in this session. And we would like to ask your opinion on these topics. And if you have your own examples or comments, it is very appreciated.
Well, for starters, is that first question is how do you actually do it or how should you do it? Because often those two are very different.
Yeah, a lot of the requests that we get these days are actually around destructive testing when you look at, for example, tissue samples and so forth, or minerals. Typically, we provide just a grain or whatever. But for specimens like paleontology specimens, we do have a process to evaluate the benefits versus the loss, and that takes into consideration a whole range of things, such as, for example, like with the deer material, where you might have a lot of it, and therefore it wouldn't, it's not going to impact your collection. Yeah, but we have a formal procedure to go through.
Thank you. And
So I think a lot of people tackle this in different ways. Some approaches are funneled by personal interactions with collection staff. Other times there might be a form that you can find on the web that's asking for different information. But like what was mentioned, sometimes it's sample size that will influence whether or not the request is honored. Sometimes it's honored with provisos or revisions in how that material is documented, whether a traditional mold and cast is made and a replica is available and shared, or if it's surface scanned and a 3D model is generated, and that then becomes available for 3D printing. But a lot of times it's, at least here at the Yale Peabody Museum, it's a collaborative process within our division where we receive requests that are provided to us, and they go through a written proposal that requests that they outline certain information, and our curatorial team reviews that, as well as our preparation staff and our collection staff. And from each of those perspectives and expertise, people will make comments and ask for clarity, or perhaps a different material would be used, but we also request documentation of that as applied. So it really varies. Sometimes if it's the only one, interestingly enough, if it's not a type specimen, that permission is granted because it will be the one used to generate new information that otherwise wouldn't be available. It depends on the locality, and they're not renewable samples in many instances, so you do need to be judicious.
And to piggyback on that, making sure you digitize, photograph, 3D model photogrammetry, something so that you're visually capturing and also potentially molding and casting. So you have, if you do have a small sample that's taken away, I think Yuri mentioned in your presentation, but just capturing as much data as you can if a request has been approved before it is sent out. Because with the question too, I receive a couple of destructive sampling requests each year, and like Yale, the Museum of Comparative Zoology also has a formalized application process. They have to state their qualifications as researchers to say they have done this kind of sampling in the past. They have publications, or if it's a PhD candidate who has an advisor who is well practiced in this, the advisor then has to prove their credentials for conducting said analysis, especially if it's something like an isotope sampling or histological sampling especially, which is much more destructive. And then that gets reviewed at multiple levels. And we have sometimes asked for them to either cut back the number of specimens, swap specimens, or outright reject it. So it is a careful analysis depending on what's requested.
Hi, I'm at the Smithsonian, and we have a very similar process, like very collaborative in terms of who's reviewing that proposal overall. For us, we've been working a lot in terms of documentation ahead of time and asking questions ahead of time, but trying to build resources that are also publicly available and externally available to share, especially when it comes to each of the different stages of the actual destructive analysis, or if someone's scanning and printing, and like any rights that are associated, if there's any sensitive need or restrictions that are needed on it, but also for molding and casting, because I think we've had a lot of those past also of people molding and casting multiple, and then we see that they're distributed everywhere and we don't know they're there. So trying to be a little bit more controlling not controlling, more observant of where they are, and just saying, that's great, let us know that you have it so we can connect it and keep it data associated with that specimen in the cast that's existing or the 3D print that exists. But yeah, I think definitely into helping and sharing any documentation or processes and templates that everyone can help utilize too.
I'm sort of curious how people have also considered, or not, maybe just PPE protocols for this too, as you curate materials in your collections and it goes through this process and develops its own history from the field to the lab to research or exhibit or display. The considerations that you're using that may affect the interpretation of results. Things like DNA, also temperature and relative humidity, how that changes once it's extracted from its environment, and whether or not we as a community can start to provide better guidance of the use of gloves, specifically in paleontology, is not that common, and yet our skin sheds cells, and that can sometimes throw a wrench into the results that we're getting that are not consistent. The other thing is the storage environment as well. When these samples are returned and the remnants come back to us, how is it that we track them and treat them and associate the results of that data with our relational databases, and how do we make that available to people who are using the collection who may be unaware of previous work on it and might be doing something completely different, but how do we elevate those standards?
At the Museum of Comparative Zoology, something that's also been beneficial is that there is a institution-level destructive sampling policy. So it covers all modern and paleontological specimens. And then additionally, because paleontology has additional destructive sampling considerations, I know my department has a vertebrate paleontology specific destructive sampling that sits on top of the museum level one. And so at the museum level, there's a minimum expectation from all researchers who are requesting and approved for destructive sampling museum-wide. It impacts all departments. So if you can, at your institution, have that high-level conversation, what do all of your departments agree upon? And you can have an institutional level policy and go further beyond that to layer up depending on if you're a department specifically, perhaps your curator is more particular or more protective in these kinds of considerations or not. And then also we have written into our loan documentation when we do approve destructive sampling that any and all notes, procedures, materials, tools, applications, preparations, all documentation, a copy is given to us. All byproducts are given to us. We retain all ownership of that material, but we do protect the data. We do not share data publicly in that regard so that their research is still protected. But we do require that we receive a copy so we can do, as like Vanessa said, is track the history of these specimens through time, and then we associate that data with the specimen records and any byproducts. If there is a physical byproduct, if it wasn't fully consumed, we can, in our database, we are fortunate to have the ability to track that information to say we have a sample that we have a histological slides, or we have a puck that potentially a new slide could be made from if needed.
I just wanted to make a brief comment about that, is that that kind of tracking can go across different types of destructive sampling. So if you've already had someone who's 20 years ago done histological sampling and someone wants to do something destructive isotopically, maybe the little remnant that's good for nothing is perfect for that additional destructive sampling. So that kind of tracking is not only good for us in understanding the history, but it actually allows us to have a longer and more productive conversation with the request generator so that we can say, well, actually, I know you want to do a new cut, but is this an appropriate sample, this thing we already have for you to use?
Yeah. So the time is now finished. So thank you so much for your active comments and inputs, and thank you for presenters and attendants.